Click for IB Biology. Here we're looking at water potential, osmosis. This time we're going to be looking at osmosis in plants. There's a blank PDF of this presentation down in the description below, along with 10 questions with answers, and there you'll also find an AI prompt which will take you through questions at different levels to test your understanding of this part of the course. So we've already determined that in terms of water potential, osmosis can be defined here as the movement of water from a region of high water potential to a region of low water potential. And I've been encouraging you to think of this water potential as being equivalent to the water molecules having high kinetic energy. And of course, as we saw earlier, this is the same as saying that there is low solute content here. The water moves to a low water potential. All right, so that will have a low kinetic energy here, and that's because there's a high amount of solute dissolved in the water on that side of the membrane that separates these two solutions. So high energy on the left, low energy on the right. That determines the direction of water. Now, what we're going to do is we're going to look at first hypotonic solutions, which we've previously looked at, in terms of water potential, and then we'll look at hypertonic solutions. So a hypotonic solution, you'll recall, is one which has a low solute content. And so this means that we will have a high water potential. The water molecules have a great deal of energy, potential energy in the water molecules to move towards regions of lower water potential. And so in our cell shown here, the external solution, which is known as psi S, so that's psi is the water potential of the external solution around the cell. And then we're going to talk about psi S of the cytoplasm, which is here. So this is still the water potential. So the water goes from the high water potential to the low water potential, and so therefore the water will go into the cell like so. Now, as we have previously discussed, this net movement of water into the cytoplasm causes a volume increase and therefore a pressure increase, which leads to a condition that we call turgidity. The turgidity and the pressure which is generated is caused by the outward pressure of the expanding cytoplasm of the cell on the wall. And of course, according to the laws of physics, there will be an equal and opposite force pressing backwards. And this force, which is additionally created because of the strong cellulose cell wall, we create what is called wall pressure, which is called psi P. So overall then, what we know is that the tendency of a cell to attract water, the water potential, is now going to be described in terms of psi S, the concentration of the solute in the cytoplasm, and the development of the cell wall pressure. Let's look at hypertonic solutions. Now, the rule remains that water will move from the high water potential to the low water potential through the semi-permeable membrane. But of course, with plants, we have the cellulose cell wall to contend with as well. A hypertonic solution, a hyper hypotonic solution has a low water potential. So let's just take a moment to think about that. If the cytoplasm has a high water potential, then this means that there's a high kinetic energy of the water molecules in that location. So it's higher inside the cell than it is outside of the cell. Psi S of the cytoplasm is greater than psi S of the external solution. And so therefore the water, let's follow the rule here, the water will move out of the cell. Now the consequence of this, of course, is that there will be a volume decrease, there will be a pressure decrease, and of course you'll recall that this leads to a condition known as plasmolysis of the cell, of the tissue. In this situation, because the cytoplasm is no longer generating a pressure, there will no longer be a wall pressure. And so psi P will be equal to 0 kilopascals. And so that will be lost. So in this case, psi P is equal to 0. We can remove that from our equation here. And so when cells are plasmolysed, for the complete plant, this leads to a condition known as wilting. As we move through the different parts of the course, looking at animal tissues and plant tissues, the application of water potential in the higher level papers will be in terms of psi W, the water potential. Links to the channel and to other playlists are appearing on the right hand side now, where you can move on to another part of the course.